Good afternoon, good evening for some of you that may be in other places of the world. So the session is a review and discussion of Global Immunology Observatory program, a weather map of viruses. And then we have a second presentation on premise, opportunities for diagnostics and therapeutics. I am Ligia Pinto. I am a Senior Vice President of the Frederick National Lab. I am the Director of the Vaccine, Immunity and Cancer Directorate at the Frederick National Lab in the US, which includes the Human Papillomavirus Serology Lab as well as COVID-19 serology laboratories. My lab played an important role in the understanding of immunity to HPV vaccines over the years, and we reached the point in serology where we felt standardization was needed, so we established an HPV serology standardization initiative in 2016, sponsored by the National Cancer Institute and by the Gates Foundation. And then that work really led to the establishment, more recently in 2020, of the National Cancer Institute Serological Sciences Network for COVID-19, SeroNet. And these efforts really have been instrumental in standardizing technologies and advancing the understanding of the immune responses to infection and vaccination, and then translate that knowledge to public health benefit. So our panelists today are, we have the pleasure to have Dr. Michael Mina and Dr. Alison Spalding. So we'll start with Dr. Michael Mina. He's a physician scientist. He's the founding director of the Immunological Observatory, which is a nonprofit working at the intersection of high-throughput immunological assay development and mathematics towards the development of global immune-based surveillance. He's also the chief scientific officer of IMET, where he leads the NIH Test to Treat program and serves as a public advisor to many different organizations and different companies. Before he took this role, he was a professor of epidemiology and immunology and infectious diseases at Harvard School of Public Health, and also Associate Medical Director of Molecular Virology at Brigham and Women's Hospital and the Broad Institute. With colleagues at Princeton, the Wellcome Trust, and the NIH, he developed the idea of a global immunological observatory that could serve as a global weather system for viruses. Welcome, Dr. Mina.
Okay, thank you. I have to hit this. Well, I'll just get started, I guess, or maybe it'll get up. So, okay, great. So what I want to talk about today, if I can get it to go backwards, is this idea of a global immunological observatory. I think in some ways the whole notion of it doesn't really need to be explained to this group, so I'm not going to go into too much detail about why and things like that. Most of the talk is going to focus here about some of the new technologies and approaches that can be built into it. What I'm not including in this talk is a lot that we've done over, we started conceiving of this now 10 years ago, developing the tools and technologies to enable it in a sort of massively throughput approach to immunology, and in a way that can extend well beyond infectious diseases. Mostly what I'm going to be talking about are technologies that are able to be pretty flexible in terms of what types of antibodies and proteins they can detect. And would love to, in the conversation that happens after, get into some more of how to actually get these types of tools funded and essentially bring them to fruition. So I'll probably skip very quickly through this initial piece because it's really talking about surveillance, and these first slides have had to be put back in because so many of the talks I give now are on the heels of COVID, where people just, with this room as a major exception, just don't believe that serology is a thing that can actually work for public health. I think people in this room, we probably drink our own Kool-Aid a lot and just take it for granted. But I start talking to DOD, I start talking to the White House, talk to whoever, and they literally say things like, serology doesn't work. And I said, what does that mean? Serology doesn't work or antibody testing doesn't work. That's really weird to say. And they say, well, wastewater is where it's at. And so I've had to go back to the drawing board almost since, like, compared to where we were in 2012, and actually start explaining why antibody-based testing has a place in public health. And it's, policymakers in particular have completely lost track of one of the core technologies and tools of public health. And I think it kind of behooves us as a community to recognize that that's where we're at when we're thinking about funding these programs. We literally have to talk about why testing for antibodies is different than testing with a PCR test, and how they each have their strengths, and how this is not a new thought, but actually this is kind of getting back to where we were. Now this is just one example. This example came from way early in the pandemic, when just people said, well, we need a PCR test, we need to know, and we went into every nursing home in Massachusetts and said we have to, between now in February and May, it was mandated that every nursing home, if they wanted to keep some certification, I forget exactly what it was, needed to test all of their participants. I said, well, that makes no sense. That's a one-time test of all of the patients in your nursing home. Otherwise, you're at risk for losing massive amounts of funding. And I said, well, if you're going to do that, then at least try to make some sense of the numbers you're going to get. PCR might give you 5%. What does 5% of your nursing home being positive with a PCR test mean? Well, if you have serology to back it up, you have a lot more information about what that 5% means. If you have 60% seropositivity status already, then the 5% is probably not too concerning anymore. But if you have 5% seropositivity status and 5% of your patients are currently COVID-19 positive on a PCR test, that's massively concerning, and you should start allocating resources to that nursing home. So trying to get serology to participate in other surveillance platforms that are currently being funded, I think, is a really important piece. So this is a similar thing, trying to explain, why is serosurveillance appropriate? Why does it have, where is its value? It's very easy, and the average Joe on the street and the average policymaker, and frankly the average physician, is going to say serology and antibody testing can't help us in this pandemic or in real-time understanding of outbreaks because there's a delay of an antibody response. That's exceedingly myopic, individualized thinking, where you say, okay, yes, it won't help on that patient, but at a population level, the durability of the signal of the antibody response greatly makes up for any lag that has in terms of becoming positive on a test. If you have if you're comparing a PCR test, which might take 3 or 4 days to turn positive after exposure, versus an antibody test, which might take, on average, 12 days. And so most people will say, well, that 12 days versus 2, I don't want that. That's not going to work out. But at a population level, the durability of the signal just well makes up for it. But developing the metrics to understand these and be able to explain them to policymakers and others is going to be really critical. This is just an example. This is from an agent-based model. We kind of simulate all the different populations in a city, drop outbreaks into it. But the question was, given different frequencies of testing, we're not testing everyone, but given different populations that we're going to be measuring on a given basis and at different intervals, what's the likelihood, what's the probability of detecting a new outbreak as it's emerging if you're comparing apples to apples and saying, hey, we're using PCR and we're trying to detect a new outbreak versus antibody-based testing? Well, as we start to get into higher and higher percentages, like pretty much untenably high percentages of the population, that extra few days delay between antibody response becomes more important. But if you're back on the left side of this, where you're testing around 5% of the population, and that's still a really high percentage of the population to test in any given interval, like every month or so, antibody-based detection actually becomes more powerful, not less, to detect a new outbreak earlier, despite the 2-week lag. And there's a lot of things behind these results. But ultimately, we end up seeing in the blue bars here that the day the outbreak is actually detected becomes faster and faster and faster as you start to get into more reasonable periods of actually testing and monitoring people. So I'm gonna switch gears. Serological assays have been stuck in what I would call sort of this very singleton, maybe handful of pathogen domain for a really long time, and that's for a good reason. Technologies are just growing up. But we're in a really different space now. And if we can work towards actually making some of these new technologies available and more robust and better in the clinic and all kinds of things, all of a sudden the question of what's the value of serological-based testing totally flips on its head. Because you can do so much today with a drop of blood, and for a cost that's less than a single Luminex assay, you could do hundreds of thousands of distinct antibody assays. We're getting the cost down to like 10, I'll show you the data, but you can get the cost to under $10 to measure hundreds of thousands of antibodies at a single time versus an ELISA, which might cost $2 for a single protein. So this is just one example. There's a bunch of different foundational technologies now. This is one that I've been intimately involved with for about 10 years called VIRSCAN. It's a FIPSeq, it's phage display immunoprecipitation. But ultimately, the way that it works is you can download from whatever database you want all the proteins and proteomes of every pathogen known to infect humans. You can throw in 100 other pathogens if you want. You could throw in the entire human proteome. We've done all of this. And you can package those proteins, get them synthesized, and package them into phage. Phage are great because they self-replicate. They're super cheap. You don't have to synthesize them each time. They synthesize for free. And once you package them into phage, which are essentially little molecular robots that present little peptides on their surface, they can each become their own little Luminex bead, their own little ELISA. You then sequence the phage that have bound antibodies, and you can get signatures like this. What you're seeing here is each column is a kid or a person, and each row is a different epitope from one of those pathogens. And there's hundreds of more pathogens, their full proteomes encoded in this platform. The density of the color is reflective, in this case, of the antibody titer, and each of those is its own antibody. So what you can see is each kid has their own antibody profile. There's some very public epitopes. Most people, if you scan left to right, there's some epitopes where most people will recognize, and then there's a lot of epitopes that only one person out of 1,000 will recognize. So you get a lot of diversity. I won't go through how we make it. And what's really nice with that diversity is you can actually use it to start reconstructing longitudinal cohorts. So this is just an example of using this technology to look at somebody's rubella antibodies. Now, each line here is a different child. This is a birth cohort. So you can see maternal antibodies waning. You can see them starting to get infections or vaccinations. But what's really important here is the y-axis on this is not an antibody titer. It's an antibody breadth. This is saying how many distinct epitopes, after we deconvolute a bit, is each individual recognizing. Each of those different epitopes then has its own titer. There's another dimension, which just gets a little crazy to start plotting it all. But you end up getting an enormous amount of data. And this is just rubella here. So this is rubella and measles. You can see that these kids clearly just got an MMR vaccine at around 500 days of life. But you're not just getting, with the same assay, you're not just getting rubella or measles, you're getting hundreds of different pathogens. And so with one assay, and less than 0.2 microliters of blood for each time it gets run, you get a huge gamut of distinct serological profiles for each pathogen for each kid. So taking this and scaling it, the value proposition of taking these types of massively multiplexed assays into the serosurveillance domain and actually utilizing them for public health really flips the financial equations on its head. I won't get too much into it, but I would love to talk about it later. This type of technology not only enables us to do serosurveillance, but it gives us predictors of cancer. It gives us predictors of neurodegenerative disease. The breadth of somebody's immunological response, the shape of that response, and the way that people respond to a regular vaccine against flu holds information about how we're going to respond when a tumor cell starts to develop inside of us. It holds information, the stability of our repertoire over time when it starts to become less stable, holds information about our immunological health, which gives us a lot more information maybe about our longevity. So this is an example from an individual taken over 10 years of time. Within each of these columns, you see EBV, RSV, rhino, flu A, flu B. This is all one person, but within each of these pathogens is 10 years of time. So each small column, it's almost hard to make them out. You can see that there's is a different point in time. So the thing to notice is that this almost looks like an individual barcode for this person that's just persisting. It's highly, highly stable. It's not just a stability, though. Here, this person got a flu A infection. Here, they got a flu B infection. We know it wasn't a flu A/B vaccine because those even though they look kind of similar, they're actually at 2 different time points. You can see that their EBV started to get reactivated at different points. The sensitivity of this assay, because we're looking for hundreds or thousands of distinct epitopes and antibodies for any given pathogen, becomes remarkably high. The specificity, too, becomes remarkably high because we have such a plethora. We can use machine learning, we can use neural networks to really hone in and get very, very sensitive and specific assays, despite the massive breadth of response here. So this is an example of how we can leverage that stability. Biobanks and blood donor centers are full of blood. Every single day in America, we throw away millions and millions of tubes of blood. Quest, LabCorp, Red Cross, Octapharma, Takeda, plasma donor companies, millions of samples of blood are getting thrown away every day. But if we could harness these fractions of blood in a way before they get thrown away into the garbage, whether it's at hospitals, clinics, blood banks, whatever, it would be remarkable. And what's included inside all of these blood donors and tubes that are getting thrown away is longitudinal cohorts. We don't normally know they're longitudinal, though, because we don't know whose blood is what. We don't know who it belongs to. And we're generally not going to, in this space, get access to that information too easily. But what does exist inside the blood is a barcode, totally anonymous barcode, that allows us to say, hey, this blood that came in today is it's from the exact same person who came into this clinic or this hospital or donated blood a year ago. There's ethical concerns here, I totally get that. But at the same time, it's completely anonymous. Unlike genetics, we can't reconstruct who the antibodies come from because it's purely, it's really random. And if you look at all these profiles that we have of tens of thousands of runs of this, everyone's unique response is quite unique. I'm an identical twin. My response is not close to my twins. It's more close than mine would be to any of yours, which is interesting, but not nearly close enough to actually infer that I'm related to my twin brother. So I won't go through this, but I do want, this is a real example. So we just took 800 samples, just random samples, and threw in a bunch of longitudinal cohorts that we had already collected. But we put it all together and mixed it up and said, can we actually reconstruct the cohorts? And actually, what you saw before is actually a reconstruction. Oops. It's a, can I go back still? I don't know. It's a reconstruction. So we started with essentially something that looks like this big blob here of 800 different samples. And what you're seeing is just the breadth of antibody titers, or breadth of antibody repertoires. On the y-axis, this is rubella and measles. It just kind of looks like a big cloud. And when we actually go and ask, can we pull out of this big cloud of specimens that are all anonymous, individual cohorts, just based on their antibody profiles alone, can we actually recreate what I call digital longitudinal cohorts? We can with exceptional accuracy. So we can also use the breadth of a response and the fact that these technologies, with some tweaking, can get down to the antibody, to the amino acid level, distinctions of where exactly somebody's antibody is binding. You can do things like alanine scanning mutagenesis and throw that into your phage display library to be able to really target exactly where that antibody is binding, not just say spike protein. Like, I get uneasy when I talk about serology to most people at this point, because like spike protein, what does it mean to have a spike protein antibody? There's tons of different epitopes and ways that those antibodies can bind. And we're talking about, what this one big average means. Well, what if we were to break that spike protein down into a whole bunch of little bits and get antibody titers for every single part of it? Not just spike, but the entire proteome of the SARS-CoV-2 virus. We can do it for the same price as one spike protein ELISA, essentially. So this is an example of a dengue proteome. And we had 30 or 40 people with Zika and 30 or 40 people with dengue. We did not change the library at all when Zika came in. We kept it exactly as dengue. This is just dengue. And we asked, can we decipher who has a Zika infection from who has a dengue infection without altering anything? Because we want to see, can we actually make sense of a new virus coming into the community? And what we found is there were no particular epitopes that we're actually distinguishing. These are dengue folks, and these are Zika folks on the other sides of the blue line. We couldn't actually see, using normal statistical methods, that there was like any given epitope that was itself going to be particularly reflective of Zika versus dengue. But when we started doing some alanine scanning mutagenesis to get down to amino acid level binding, and we start putting it into neural networks, I'll just fast forward and get to the chase here. We started just with our first neural network that we ran, this is a convolution neural network. We were able to get pretty darn close to 100% discrimination. But with just adding an ensemble to this, we were able to get to 100% discrimination. This is out-of-sample discrimination, just from training on, I think it was something like 30 or 40 samples of each, known Zika and known dengue. This would have been an important tool at the beginning of Zika when it took the major manufacturers like Abbott and Roche and some others. So, I'm forgetting who was building out the Zika antibody tests. It took almost a year before a commercial assay came to fruition that was actually able to reliably distinguish Zika versus dengue. That had massive consequences when somebody went on to get dengue. The questions were, do I need to abort my fetus? You know, these were real questions people were asking, but we didn't have a solution because diagnostics took too long to build. So getting back to the Immune Observatory, and I'm just going to kind of give this high level and then stop. The blood can come in from all different ways. Yes, we and lots of people in this room have worked on the statistical evaluations of how to make sense of the different biases that come in from different groups. Super important, absolutely essential. But the takeaway here is there's lots of different ways to get blood, and none of them necessarily need to be dedicated to serological surveillance. They can be using discarded blood samples, leveraging pharma companies that want to really be driving vaccine trials and want to figure out how to get the most bang out of their vaccine trial as quickly as possible. A company like Pfizer has billions of dollars literally on the line if they could speed up a trial. So they'll pay for surveillance programs that would help them better understand where is the virus of interest circulating, or where is it most likely going to be circulating tomorrow, so that they can actually define where they're going to put their trials. So there's tremendous numbers of ways to fund this that fall outside of the ways that we normally think in this community. And so we actually did a pilot using Octapharma. Octapharma is a great example. They're a big plasma donation company. They actually pay people to go donate their plasma so that they can then fractionate and sell it to pharma. So that it's big money. We were able to process over 1 million samples, 1 million, working just with this one plasma donation company that was constantly collecting blood, paying for it, and then they paid us to do it. We weren't trying to go to the government and try to figure out how to get it funded as a research program. They were willing to give us the money, and they funded all the logistics. And these are just like massive freezers because all the restaurants in Boston closed down. We needed to put it all in freezers during the pandemic so that we could actually figure out how to like get our high throughput equipment, going fast enough.
That would never happen at McGill.
It would normally never happen at Harvard either, but that's what money does, I guess. So I'm just going to show one more slide, I think, but we can do it with saliva. We've done this where you can just rub a swab against your gums, and you can get almost a full representation of your antibody repertoire just from rubbing somebody's gums with a little sponge and sticking it in buffer or saline, just like we do with blood. So there's lots of new ways to do it. I think that it's highly scalable. The benefits of doing massively multiplexed assays goes well beyond just serosurveillance. And if we can tap into those other things, whether it be autoimmune disease, cancer, neurodegenerative disease, areas where there's just billions and potentially trillions of dollars, like literally trillions, like a GLP-1s, trillions of dollars are on the line. If we can tap into that, we wouldn't be having discussions about how to like fund $20 million for a serosurveillance program. And I really, so the hope here in showing this is like, I see this as a future approach to actually performing assays in a way that can extend way beyond infectious diseases. And then hopefully those of us with interest can just pull some of that data in and leverage it but really get it into the places where the money's at. So I'll stop there.
Thank you so much. We are moving to Dr. Alison Spaulding's presentation on premise. She was introduced earlier today, but I will say that she is the senior advisor and team lead at the NIH NIAID Vaccine Research Center. And today she will be presenting about premise, a program that she's leading. Thank you so much, Dr. Spaulding.
Thank you. Let me make sure here, do I click? Oh, just forward, okay. Well, good afternoon, everyone. Thank you so much for having me. I'm very excited to be here. It's been a wonderful meeting so far, and I can say that I really think that premise is an opportunity to partner with HemaNet and a lot of you here because our program does not exist without samples. It does not exist without access to biorepositories and the kind of work that HemaNet's talking about doing. So I think that this is a really good match, and I'm looking forward to telling you a little bit more about our program. So as was mentioned, we're based at NIH. We're with the Institute of Allergy and Infectious Diseases, and specifically at the Vaccine Research Center. We're an intramural group as opposed to the extramural group, which most people are familiar with, which is the majority of NIH's funding. But we are an entity within that, and we focus on the research in many different infectious diseases. The PIs that are at the VRC cover, as you can see, are a number of different agents and viruses. But our work is in pandemic preparedness. I did want to take a moment and just explain, though, how the VRC is unique within NIH, and it really operates kind of like a small biotech company in that we have the capability to bring products into phase 1 trials. And so that begins with our group, our basic and translational group. And so a lot of the premise work is embedded within that group in particular. That's on the main campus in Bethesda, Maryland. We then have a facility that does the vaccine production. That's located in the suburbs of Maryland as well. And we work closely with this group as well. We then have a group in Frederick that does the vaccine and monoclonal antibody production for us. And then those products are brought back to the clinical center at NIH for phase 1 trials. And so, again, the kind of uniqueness here, in addition to the kind of the high throughput assays that are involved in once the trials are underway, it's really a unique opportunity that we have to be able to translate products into potential market products. Now, I will say premise as a whole, we, and I'll talk a little bit about this, but we don't necessarily envision that everything we work on will be carried forward into clinical trials by the VRC, but we do have that capability, and especially as we address some of the products like was discussed this morning when there is a little bit more of a challenging market, a commercial market or a limited commercial market, the VRC really does have that ability to move products forward that might not be otherwise of interest to pharmaceutical companies. So premise really came out of, as we've been around about 2.5 years now, so obviously we came in after the emergence of the pandemic, but we really wanted to kind of answer this question and fill this gap of the immunology, which of course is what we've been discussing a lot today. And this group here is well familiar with why this kind of work would be necessary, but we believe that it is actionable, and that's kind of how premise came about. So in regards to what we are specifically, we are a pathogen agnostic, immunologically focused pandemic preparedness program. And our goal is to establish a preemptive repository of immunobiological countermeasures. And so the idea is, again, that we have kind of tools on a shelf that could, again, either be moved forward by us or be moved forward by some of our partners into clinical products. And we're really focused on 3 publicly available deliverables. That would be reagent data resources for diagnostics, monoclonal antibodies with potential for prevention therapy, and vaccine immunogens. And really the idea here is that we want to make this work publicly available as quickly as possible. We can discuss some of the challenges in doing that, but that is the goal of the premise program. So we get a lot of questions about what is premise not, and so I just wanted to differentiate for a moment. So as I mentioned, we are pathogen agnostic, immunologically focused, and we're really looking at the translation of surveillance into clinical products. So we are not a serosurveillance network. That's why I was saying we need, though, networks to fuel our work and our progress. And we are not an early warning system. We are not quickly set up for anything like that. And so just wanted to highlight that. So wanted to go through how our workflow works. So right here, right off the bat, we see exactly why something like HemaNet would be critically of interest to us. So we need samples to do our work, and in particular, we need PBMCs. And so that obviously is a unique challenge, but it's one that we've been quite successful in overcoming so far. We then have a group that does virus discovery, and by group, I mean 2 people, we're very small. that does down selection and antigen design, and then that informs our serology that's done in-house at the group that I mentioned that is in Gaithersburg, Maryland. We then use the samples, obviously, to do serology and B-cell work. This is then used to inform the monoclonal antibody development process and the functional analysis to get to our outcomes. So this process here, our program director, Danny Dewick, who's one of the PIs at the Vaccine Research Center, has a process, a rapid monoclonal antibody identification process that he affectionately calls RATPIG. And that, I wasn't consulted on the name. It takes actually only 6 days to identify upwards of 500, sometimes 1,000 monoclonal antibody targets. So I'm very excited that in this workflow, we're actually getting to that point where we've started to do some of those sorts, and so I can talk about that. But it's been a very exciting process to be a part of. And then we have partners such as Lawrence Livermore National Labs in California that we're working on antibody optimization with. So as I mentioned this morning when we talked, we have 88. I think now it's 89 because we subscribe to ProMed, and so I'm constantly adding viruses to our list. But these are the viruses that we intend to target. I mean, it's a lofty goal, but strategically we do hope to get some kind of something on the shelf for all of these viruses over time. And as you can see here, obviously we prioritize those that are already known to be human-to-human transmission, but we have zoonotic ones as well. Our main technology for serology is the MSD plate and MSD technology. And so what we've done is we work with them to develop customized assay plates. But I did want to let people know, because I know there's some people in the room that work with MSD, we are working with them to make this commercially available. Which is kind of hilarious because we get absolutely nothing from it, but great for them. We're like, you're welcome for helping you do your job. So this is the first plate that we helped to develop. This one we have been running samples on. Things are looking great. The plate is working very well. And this is based off of an observational cohort study that we have ongoing. I'm not going to talk a lot about that, although our colleague Daniel Park is going to present after. He's been doing all the data analysis for us for that, but that is an existing cohort study we have going on looking at children in the US. And so obviously you can imagine that's why these viruses are on this panel. But then we're about to get printed next week, actually, our first regional panel. And again, these are developed in cooperation with our strategic partners that we work with. I'll talk about who they are in a second. But it's a very exciting part of the job to ask them, like, what are you concerned about? We'll put it on a plate for you. So that's our Africa and Middle East one. We then have up next the Asia and Australia panel, and then the Latin American panel. We do have in-house capability for protein production. Again, we have 2 people. So we do rely sometimes on some external partners as well to do protein production for us. So you're all familiar, of course, with the virus world domination plan. But we also have a plan. So this is our current network. And a lot of what my job entails is building this network. As you can see here, we do not have a Canadian site, so I'm looking for a partner. And I'm sure that I will find one here. But as you can see here, we have ones that are in development and ones that are currently established. What we do is we actually have research collaborative agreements is the main document we use to establish our partnerships. And we've had a lot of success. It's mostly lab to lab, I'll be honest. We definitely don't work with large organizations. And these are mostly academic labs, some government as well. But again, it's kind of mostly just kind of that peer-to-peer work that we're doing, informing these. And so, like I said, there's 6 of us total, so it makes it sound like we're a lot of people. We're not. We're real small. But this is a list of our domestic and international partnerships, and we have, again, 14 international and 10 that are pending as well. So that also is never a dull moment. So just in brief, kind of how we engage with partners. So we offer to our partners the ability to kind of take advantage of our technology, to see what we're doing, to learn about what we're doing. We are constantly training people in our lab, especially on the RATPIG technology. And as far as what partners can provide, samples, access to samples, as I talked about this morning. Maybe it's that you don't have samples, but you have an ability to call people back, and you didn't get PBMCs originally, but you maybe could. That's something we do a lot with our partners. But given that we're not extramural, we don't have any ability to provide our own funding. But again, we do support our partners in different ways to do the work. So I'll just end with this. This is just some of our current projects, just to give you kind of a flavor of what, what's to come in our pipeline. So I already mentioned the EBD68 focused pediatric cohort study we have ongoing. We're working with the CDC on an Mpox specific assay. And then we have a number of different diseases that are in our pipeline that we're developing neutralizing antibodies for. And these are all done in conjunction with our key partners. So as you see here, Langia virus here, that's with our colleagues in Singapore. CCHF is a collaboration we have with Rocky Mountain Labs, although once let's say if we were to get to the point of any kind of human trial, we would obviously be looking for an international partner for that work. We're looking at Junín and Machupo and Oropuche, and then like SFTSV is a perfect example of how I'm really hoping our collaborations go. So we have samples that have been sent to us from our colleagues in Korea, and they're coming for training actually next week. They're going to learn the RAP technology, take it back. My hope is that they will actually be the ones to move forward in any human clinical trials because it's endemic in South Korea, not obviously in the US. And so that's what I'm hoping I can kind of push forward with them to be the ones to get that product to market. We work on Lassa virus, as I mentioned this morning. We have a very extensive collaboration with Sheba Medical Center in Israel. We do a number of different things with them, and in particular, West Nile is a big focus for us with them. And again, this is where we are very flexible. What do our partners want to do? What are our partners' needs? What are the country needs? We're able to kind of pivot that work. Very excited about a lot of Middle Eastern studies that we have going on, looking at Hajj, people before and after going to the Hajj pilgrimage. And then, as I mentioned, we have a lot of different training for RATPIG. So I will end here. Here's our team. Danny Dewick on the right here is our program director, and this is our amazing team. We would not, and I know I said there's 6. We only have 6 employees, but we have a bunch of people who help us out, and I always like to give kudos to them because we couldn't do this work without them. So thank you.
Thank you so much. And I will go into a couple of questions first. And if you both could reflect on what are the major technical and logistical challenges in first establishing, but these initiatives are established now, and maintaining them, and then translated to that public health impact as well as development of clinical products? If you could discuss a little bit about that.
Sure. There's, the technical challenges are across the board. There's going to be, it really depends on the product that's being developed, but getting it into proper facilities. So much of this work to date has been done in academic research labs. When you get out of the academic world, you realize that academics have almost no experience in what industrial enterprise type efforts look like in terms of quality controls, checks, even just software and people management and record keeping. And so those are some of the biggest things. And I think getting, one of the overarching things, if I could say, okay, if we could really get this, could it accelerate the whole field of science, getting translated, would be giving PhD students and postdocs and professors actual training in everything from, how to actually develop products that are going to be placed into people, are going to be used for diagnostics, across the board, or that are going to have to be regulated. That's a whole science unto itself that even companies generally underappreciate, and then they're 3 years past, and then they're closing down because their VC people don't want to hold on. So and most of those are run by academics initially, and so having that type of training, I think, is kind of like this base layer of what could be done that would be really interesting to get into the academic world in a much more profound way. And that's not really the question you asked, but that's my answer.
Thank you. 100% agree with that. So I would say big picture, not a surprise to anyone in the room, but the lack of or the declining interest in pandemic preparedness is our #1 enemy. Not only from a financial standpoint, just from understanding why we're doing what we're doing and why it's important. I'm sure all of you have had the situation where you go out in social settings and I tell people what I do, and multiple people be like, but it only happens once every 100 years. Why would you need to work on that? So I think that's a huge issue. And then, I think the other issue for us, and it came up this morning in the panel with our colleagues from WHO, but the market issues are a concern for us, and we're working on things that there's probably going to be limited industrial or financial interest in pursuing. and just how do we overcome that? And it's not that it's not overcomable, but it's a challenge. And again, it's in the back of my mind because we're just kind of full steam ahead on trying to get products developed. But as far as how to get them available to people on a wider scale, it's going to be a challenge. And then I would just say in a smaller scale, just the technical challenges of getting cells, keeping them stored, getting them from our partners. That's just going to constantly be a challenge for us because it's what we need for our work, and they're hard to get. So.
Thank you. And then another question, one to each program. So, Dr. Mina, so from beginning conception all the way to validation of your technologies, because it's extremely complex to validate this type of technologies for the end use, how long does it take?
Well, if you're going way back to sort of the initial conception of, okay, how are we going to reimagine how to build out a super highly multiplexed assay? I mean, that's years and years and years. But just getting now into validation, I think it's a massive challenge. And are you validating for an actual clinical diagnostic, or are you validating for public health use where the result isn't going to be used for care? Totally different, requirements in a way. But my answer to that is take it, piecemeal. Find what I would really like to do with these types of tools to get them out into the commercial market is start finding a few analytes that are of interest, whether that's of interest because the Red Cross has to measure them in every single unit they ever collect. Find a few analytes that can be really well interrogated. Remove 99.9% of all of the other analytes that are being identified at the same time, but really just focus on making sure that that particular handful of analytes that you want to get cleared through the FDA are super high accuracy. And then still run the rest of the panel along with it, but validate the rest as you go along, sort of, what's your next pathogen of interest. So I think when you're talking about hundreds of thousands of analytes, it has to be stepwise, and some of them will never get validated. They'll live in the public health world, and they'll be validated here and there in different laboratory research projects, but never in like a super robust way because there's just too many to make it feasible. But I think just really identifying what are the ones that are going to be financially make sense to validate in the most robust way first, and then keep kind of marching down that path to keep growing where you've really done like the most robust validation.
Thank you. And then within the premise program, you mentioned that your goal is really translating to products, clinical products. Can you tell us a little bit about your top candidates? I don't know how much you can disclose, but where are you going into phase 1, phase 2?
Nope. We're pretty much further back than that. So of our top 6, and they're the ones I mentioned on the last slide, I would say the West Nile monoclonal process that we're involved in is probably the furthest along, but it's still still basic science. We're not at the point of having any kind of product that would be available for human trials at all. So I would say that's our most advanced, and then on our top 6, then it's SFTSV, Lassa, Hunin, CCHF, and Langia. And again, that's an interesting mix because Langia virus has only had a handful of human cases to date. So that one's more proof of concept for us. Like, can we take just a sequence and get to a monoclonal antibody candidate, as opposed to because we had no access to human samples. And then whereas like something like West Nile, obviously we have a lot of samples that we've been able to use from humans. So to be continued.
Yeah, lots of work. Thank you so much. Amazing. I would like to open the discussion here in the room. Any questions or online?
Okay, I'll go before Tim because he's tougher and will have these really great questions. So I have a couple of comments and then some questions. One is, the FIP-seq technology, I think really is that wonderful technology to be able to inform of which epitopes should go into these serology tests. So really want to highlight that we should be thinking of, if we can't use that readily in our serosurveillance work, we should work with partners to understand how to build our serology assays that way. And so I think that's really important. The first thing you said, Michael, though, or early on, was sort of, were not viewed as maybe important, the serology world, and we got drowned out by wastewater and stuff. And I think it was Morshid or someone. I didn't even realize that was a joke, drowned out by wastewater. I'm going to use that now. I think Morshid once told me, too, there's something about being a squeaky wheel. And specifically for FIPSeq, when I went to the ASM Microbe Conference in 2019, the best talk I saw was Charles Chewy linking D68 infections to the neurological conditions, and it was a fireside chat of like 10 people. And so, you know, being at conferences, putting the sessions together, being loud maybe for us to work on. So then now I'll get to my question. And I want to commend both of you, though, because I think the work you're doing and the partnerships you're making with folks like us in public health labs to give us those technologies are just fantastic. And as someone who likes to get access and approvals via phone calls and emails, I did actually email Steve Elledge in 2019 at Harvard and asked for the VirScan library as a new clinical microbiologist. I'm telling this because I'm selling it to everyone. And he said, well, what do you want to do with it? I said, you know, XYZ in Canada. He said, sure, here it is, and sent it. Now, building that expertise in our capacity and the scalability has been very difficult. But the use case for it is in virology is the alternative to metagenomics. On a consent-based clinical use to identify etiology of post-infectious conditions, which we talked about. So very valuable and starts to build expertise. The question then is, although we're Hemonet, and so we're talking about blood, you both presented these really nice panels, or how you can start to develop panels that can be useful for other sample types. You mentioned the saliva, but CSF, I think could be, you know, is another valuable thing. As someone who, again, has more of that clinical work, so can you comment on the panels and the work that you're doing in that sphere? And sorry I took so long, but it's just such a great session. Thank you.
So on the long-term pathogen consequences, for example, I'll get to the CSF question. These tools are amazing for it. It's actually how the reason I first got into developing high-throughput arrays was to uncover that measles immunological amnesia, you know, to show that measles depletes immune memories. Prior to that, we had only uncovered it from mathematics. WHO came and said, well, prove it, essentially, you know, before we even think about changing vaccine schedules and whatnot after measles. And so, you know, that was a very important use of it, where we were able to show that measles is wiping out people's immunological memory. Same thing with EBV and multiple sclerosis. We used it to link those two. And so the potential opportunities that go way beyond serological surveillance, I think you're absolutely right, are huge. For a CSF, I mean, at least my quick answer is like it's a no-brainer. It works really, really well. If you're looking for antibodies, if you're looking for other sticky things in CSF, you have to either change from a phage display system or find, you know, the right epitopes that you want to plug into the phage. But you can do that. Phage, the technology's gotten a lot better. You can now plug in, you know, 500, 600 amino acid sequences into phage, and they fold pretty well. They're not going to have post-translational modifications and things like that. But there's a huge assortment of other technologies that fall in the same family of very multiplexed arrays that I think don't just need to be showing epitopes that antibodies bind, but any number of other detection systems.
Yeah, I'll just say briefly, we have not used the platform at all for anything other than blood, but would definitely something we would consider. And then for our EBD68 study, again, that Daniel's going to talk about next, we limited it to blood collection for the first cohort, and we had some nasal swab collection, but we actually added that as a mandatory component for cohort 2 and also saliva collection. So we are branching out a little bit, but we're pretty focused on blood for now.
Next question.
Yeah, I have one question for both of you. So Michael, when I'm in the situation where I see that Gates and others are just funding Luminex right now in big time, and then what you suggested is kind of the next step, but when and how would this be practically implemented, where that the funding that is available now is kind of the last year's method? And is it practically already to implement that phage display array, and how would it look like in real life if we wanted to run it in parallel and see and try it out? And what are the next steps?
Yeah,
for Alison, it's a different question. So if I would try to pitch this to partners in the South, what is their advantage? What, like, if they give PBMCs? I didn't hear that clearly, sorry. If you could just give that pitch again.
So the platform in its current format takes a lot of expertise, for sure. We've trained tens, or probably in the 10, 20, 30 range of different labs to utilize it. Half of them fail because it is pretty complicated. So a huge component of what I'm doing with the Immune Observatory is dedicated towards taking these technologies and trying to figure out how to make them much easier, much more like a Cepheid or a Luminex type of approach. For these particular technologies, there's a very clear avenue to get there without raising costs a lot. You know, especially in the world of nanopore sequencing, because the readout of phage is a sequence. There's a very clear line of sight to doing it. But like you said, for various numbers of reasons, there's like a huge amount of money that goes towards Luminex because it's historically been used for some time now, and it's supporting, you know, they want to be funding action versus some of the development of the new tech. We did just put in an application to DARPA yesterday, ARPA-H, a few months ago. And so we're, you know, getting it out of that academic or that sort of single super lab kind of mode and getting it into a mode where it can easily be ported into any public health lab, I think is 5 years away, and probably $100 million away. Not $100 million to purchase instruments, but I think it's gonna, you know, take about that.
Yeah, and Thomas, thank you for the opportunity actually to clarify this, because it's very important to our program. And although we are building things as they're moving and happening, the foundation of all our relationships is a research collaborative agreement, and we put together with all our partners. It's a legal document that they sign from their institution and our institution, where it clearly outlines that we are in this work together and that we are partners in the work, so that anything is produced is their access to that partner. And so the goal for partners in other countries would be, if they provide samples, they're going to have access to all the findings. They're going to have all the monoclonal antibody sequences, for example. So if they would like to move that work forward, if they'd like to try and pursue human clinical trials, they can do that. If we find an industry partner here in the US, though, that's involved, they're going to still be involved directly in that work. And because the key is we want these products implemented in those countries. And so that's why we're trying to target viruses of interest to them. And so, again, we really view this as a very strong partnership between us that has to be a benefit to our partner, first and foremost.
Next question.
Great, thanks for terrific presentations. We've sort of done, you know, national uses of serosurveillance. We had the cross-national studies, and now we're seeing where serosurveillance can be part of a much bigger global enterprise in these two cases, which are really, really interesting. So with a global perspective, I'm just wondering if you, both of you could sort of opine a little bit on where's the governance for this research that we need to perhaps accelerate the opportunities. And not to take anything away from what you're doing, which I think is great, but I sort of feel like we're missing a bigger agent who can actually enable this more significantly. So if you could just think about that a little bit. One of the things, Allison, when you were presenting, I was thinking, well, when we set up CEPI, the Coalition for Epidemic Preparedness and Innovation, 7 years ago, that was, or 6 years ago, that was really sort of the opportunity to perhaps pick up on things that got into phase 1. And they had their epidemic list of, you know, globally orphaned viruses that were of pandemic potential, because they don't fit into R&D priorities of pharmaceutical companies until they really go viral and have markets of billions, and then everybody's interested for the time that they're there. But what's that interface look like? And Michael, you've been pushing around, and you're saying that the academic environment has not been sufficiently permissive or supportive for the scale of work that you'd like to take the GIO to. What sorts of things should we be thinking about? Should We have, in the context of this pandemic treaty that's being negotiated at WHO, some space for this frontier science. Should our national institutes, we call it the Canadian Institutes for Health Research in Canada, should they, in their infection immunity groups, institutes, should they be advocating for sort of how we can harness this potential much more actively? I'd just be interested in hearing a little bit from you on whether there's some things that we're not looking at in the broader environment or that we should be looking at, which might make a difference to sort of seizing this global potential a little bit more quickly. Yeah.
So on the who should be responsible and like a governance, I think that we have a fairly decent playbook that's starting to be written, in the sequencing world. And obviously that was a bit there before the pandemic. With the pandemic, it has massively accelerated. It's the question that comes up first anytime you start talking about doing anything globally that's going to be collecting data, in particular data from humans. Like you could say a viral sequence, there's almost a little bit less concern around it because even though it's coming out of a person, and if it was an FDA diagnostic, they would still consider it part of the human sample, but the sequence itself is the virus and not the person. So, but I do think that it's more of a social thing, if we are okay with sharing antibody-based data, for example, and immunological data, I don't see it as particularly intrusive if it's truly completely blinded. I mean, you could even pull it apart and give it in different ways so that it really can't, like no single repertoire. You wouldn't really want to do that because you lose a lot of information, but I think it's, we do have to get over this hurdle if we want any opportunity for working together globally, but there's not a lot of good examples where the world really does share its data well. So it's a huge question. It's way above my pay grade to think about, but it's something that I'm forced to think about whenever I go and present it. Briefly on the kind of just for you in the last set of questions you just asked, the theme of it was academia. I mean, I think I can just say from my experience being in the academic world that academia lives in the dinosaur world of money. I mean, it just does. Industry has tons of money and academia barely taps into 1% of it, if that, and even at a place like Harvard, which has $60 billion endowment, it doesn't flow to actually do anything. And as long as that continues to happen, more and more research is going to get moved into the biotech sector and industry, which I think is totally fine. But you do end up losing some engagement there. There are really different opportunities, and you see it all the time, especially in the US with nonprofit hospitals that are revenue-generating hospitals that effectively are academic institutions that make tons of money. Somehow on their records it makes it look like they don't make money, but they do make money. And so creating revenue-generating labs, like a lot of people here have labs. Laboratories are some of the most valuable real estate you could possibly imagine. It's a lab with expertise. And if you can turn that around and use a portion of it to bring revenue, that can totally change an institution. I'll just give an example. I don't know if they'll be happy for me saying it out loud, but I'm not there anymore. The Broad Institute, I tried to start a PCR operation when COVID hit and the serology stuff at Harvard. They pretty much said, no, we don't want anything to do with revenue generation, that's not grants and whatnot. So I went to the Broad Institute, started a large PCR testing lab there. 4 years later, they probably made $2 billion profit off of that lab. I didn't see a penny of it myself personally, but $2 billion because they decided to dedicate their resources and use it for COVID testing. Now, it's an exceptional experience, but it does speak to the preciousness and the value of these spaces that a lot of us in this room have ownership over.
I'll just be brief because I know we're out of time. I would say catch us 5 years from now, right? Because I think right now for us in particular, we're in just kind of heads down in the lab, getting the work done, trying to get proof of concept that what we're talking about that we want to do, we can do. And in the 3 years, since we started, yes, we've moved along significantly. But as far as eventually, should we still be housed like at the NIH? Probably not, right? We should be housed somewhere else in a larger institution with more than just 2 people to do protein design, right? I mean, so there's that. And then in regards to like CEPI and partners like that, my hope again is when we get closer to a potential product, that's when those relationships would be engaged.
Thank you so much. Excellent presentations, excellent discussion.